All right, here we go with the last section of notes from Chapter 1. Some concepts related to doing studies in biology or experimental design, data collection, data analysis, that kind of thing. We will have a little bit more on that when we talk some statistics with you, but this is just some intro stuff from Chapter 1 that hopefully some of it's review, but if not, take note of anything that seems new and unfamiliar. So whenever we're studying living things, expect the unexpected. They're not always easy to study because they don't always respond the way we think they're going to. They could be unpredictable. They don't always want to be studied. They want to hide. So can be challenging, especially with animals. But it also makes it kind of a fun challenge too. We also have to think about ethical considerations when studying organisms, depending if we're doing experiments and manipulating variables on organisms. What is or isn't okay. And so if you go into a field of biological research, you'll work with folks that have to make these decisions in their planning. And anyway, just want to bring that to your attention if you hadn't thought about it. I hope that you're able to see that idea play out in Henrietta Lack's reading. Another challenge with studying biology is sometimes the processes we're interested in understanding happen over a fair amount of time. So for example, if you are looking at succession in ecosystems, that takes a while to get long-term data of changes in plant communities over time in an ecosystem. And oftentimes studies can be planned and actually overlap with different researchers taking the helm of the study because it might outlast the researcher's lifespan. Think about sea turtles or giant tortoises. Giant tortoises can live longer than we can. So if we are to study them, we can continuously collect data about their longevity, but we have to pass on the torch to someone else. So sometimes a little time consuming to get answers, not like physical science or chemistry where we can get, or physics where you get results very quickly. Whenever we are collecting data, so you'll be collecting data for your birds, the question is, what is sampling error? Because there's always potential error when we're collecting samples. So sampling error relates to some difference between the results, the data you collect from a small group as compared to the whole. So let's just say you were going out and measuring the wing, or not the height of, average height of men in a building. And then you would collect that data, get an average, and then you would say, oh, the average is 6'5". And then you would think, what's wrong with this picture? You know, people aren't 6'5". But if I told you that this was the Timberwolves locker room, then you would realize that, oh, our sample isn't random. And so if we don't randomize our samples, we sometimes can get error in the subset of organisms that we study. So we want to try in science to avoid sampling error. Randomizing our samples is one way to try to reduce that. also, when you just do a subset of a population and it's not random, it can prevent us from making broad generalizations about the population as a whole. So for years and years and years, drug studies or health studies were done on people, and then they started to pay attention. Who were they actually collecting samples from? Men. And so do women respond exactly the same way to drugs and medicines? And the answer is no. We have slightly different metabolisms. And so if we are trying to understand a disease and response to a disease through medication, we can't lump necessarily everybody into one group. And so when we talk about generalizing, we have to make sure that we can generalize, and we need data to support that fact. And so we need to get everyone included in the studies. Here's the biggie. Small sample size increases sampling error in experiments. So if you go out and collect bird data for an hour and have a handful of sampled birds recorded, most likely you have high sampling error. Researchers are going to be looking to... Sorry, we got interrupted with some homecoming discussion next door. So as I was trying to say regarding your sample size and bird experiment, if you were to hope to get statistical significance on your data, scientists look for a sample size of at least 100. You may not be able to do that in the time that we have, but you're always going to try to maximize the amount of samples that you can take in the given amount of time you have. So keep that in mind as you're doing your research on your lab and trying to collect data. More in sample size is better. And we'll talk about why that matters with statistics a little bit later. This picture in your book, Chapter 1, gives an idea of what they mean when they say sampling error. So if you have these green and black jelly beans in a jar and you reach in and grab one and you take a look and you say, oh, green jelly bean, there's green jelly beans in the jar. You could generalize and say that they're all green jelly beans, but obviously we know that there's two kinds of jelly beans. Now based on the percentage of each in there, if we collect severals in a sample, so pull out 10 or 20 jelly beans, we're going to get a closer approximation of what's actually in that jar of jelly beans proportionally. And so again, increasing that sample size is going to get us closer and closer to the real truth in the data. Probability is, again, something you probably already know, but the likelihood or chance that some event or outcome will occur. So some sort of measure of how likely something is to happen. And it is also depends on the total number of possible outcomes. So whenever we're analyzing experimental data, especially if we're doing statistical tests on that data, we're going to be able to calculate probabilities from that. So when you think of flipping a coin, it is possible to calculate the probability of unexpected results. So 50% chance that you'll get heads, 50% chance you'll get tails. All right, so I mentioned this notion of statistical significance. Something that we want to learn about and understand when we are analyzing data. especially when you start looking into primary literature or primary journal articles a little bit later in this class. Statistical significance is defined as, or means, that a result from data collection is statistically unlikely to have had occurred by chance. So then, if chance was not a possibility in the outcome, then we would say results were statistically significant. So statistical significance in relationship to data or results from an experiment always linked to some statistical test being run on that experimental data. You have probably never run statistical tests on any of your data ever, but we will talk about two in this class. So when we're doing science, when we're doing experiments, every result, even statistically significant results, do have a possibility of being shown or disproven or said to be incorrect. And when we are setting up experiments, we also don't want to say we have proven. We can say the results strongly support a hypothesis or an underlying theory, but they can't prove with absolute certainty, because science wants to design experimentation in a way that allows us to correct any mistakes that we might have made if we had more and better data, more evidence. Variability is something that we can have in data. So what does variability mean? Well, this dot here represents average, and variability is the difference from the highest to the lowest. So of these three, they have different averages, but they also have different variability. This has high variability from the highest to the lowest. This one has the middle amount of variability, and this has the least amount of variability from the average. And then bias, we have to also in science, in any research, be aware of bias. And I hope that you're also identifying bias when you're reading Henrietta Lacks. So that's some preconceived idea or expectation that of course shapes the way we view the world. Our worldview is tied to our bias, what we believe and value personally. So in your book there's a section that talks about a study on olestra and potato chips. And so a researcher believes that olestra causes intestinal cramping, so we'd want to reduce bias in the data interpretation. If the researcher already has this assumption, this bias, they could hand their data off to someone who doesn't know what the numbers are referring to and have them interpret the statistical data and draw a conclusion without knowing what it's about. Tell if it is statistically significant. That reduces bias. And so the scientists could hand that data off and it would be considered a double-blind study. So not only is the researcher not crunching the numbers personally, but they're also getting participants to not know which test group they're in. Are they getting regular chips or the chips with olestra? If you're not sure what the heck I mean, go read that part in the book. And then I'm going to throw this up so you can reflect on this before our time runs out. But when it comes to nature of science, we understand these things about the nature of science. And we're just hitting the tip of the iceberg, but it's an amazing process, and we also want to appreciate it in all its complexity. Here ends Chapter 1 notes.